You're on.
All right, so today, Lonnie, he just got done finishing. Today we're going to try to restore soil biology with what's called Protozoan Infusion. And we're going to apply that over here in this construction area where Lonnie just got done using the broad fork to bust up the compaction. So this has been brewing for a couple of days. It's water, it's local biology, it's a food source for that biology. And I looked at it yesterday with the microscope, and today we're going to put it out. Each milliliter of sample had 1,560,000 flagellates in it, so it's really rich with protozoa. I didn't see any amoeba in it, but that's okay. That's what the compost is for. I also didn't see any detrimental organisms, no molds or mildews or anything like that. It's just really nice, high diversity of flagellates. So what we're going to do, we're going to use this watering can, and we're going to pour that into here, and we're just going to sprinkle it out on there. And what that will do, that'll reseed trophic level 3, which will kickstart nutrient cycling. So, and it's been oxygen, you know, this oxygen source here. It's been oxygenated pretty well. So I should filter this, but I don't have a filter at the moment.
So tell everybody about that water and everything that's actually went into that recipe.
Okay, so we seeded it with a cup of finished compost, vermicompost.
Right.
We let that brew for 2 days, and at the same time we put in 1 drop of honey. And then when we assessed the biology, it was still kind of low. So then we added 1 cup of compost from our pile here. And that should have put in a bunch of amoeba and all kinds of other diversity organisms.
Plus a drop of honey.
The honey's a food source for the bacteria. So flagellates eat about 10,000 bacteria per day. So the bacteria has to have a food source. Their number one food source is sugars. The number one food source for flagellates is bacteria.
But they never get fat.
That
we can tell.
That I can tell. I don't know. They may. No, they don't get fat. All right, so we just filled our can up here with our Protozoan Infusion. Now, since this is busted up, we have a rainstorm coming about 5 o'clock today. And I'm just going to walk around and reseed this area with flagellates. So this is called a soil drench. I'm drenching the soil. Now they reproduce, so I don't have to be accurate. You know, you hear a lot about precision agriculture. These are living organisms. They reproduce, they move themselves around. So you start with a little, you wind up with a lot, and they'll be here for years and years. So,
okay.
Now it's possible to kill them off. You know, you use chemicals and salts and fertilizers, and you can kill them off, and that's what's happened in a lot of our ag soil. Back here we had some yard construction. It's damaged the soil, and we're just trying to restart biology. So you can see it. That's all it takes. The secret is, you know, knowing how to do this. And that's what I went to the Dr. Lane Soil Food Web School to learn how to do. How do we restore the environment? How do we restore soil health? And this is a demonstration. So you can see how muddy this is. This has been the 3rd year of non-recovery. Last year, this whole area here was just pure mud. There's a video online showing how muddy it was. That was seeded. It set for 2 years. It never took. So I put out some biology there as well.
Can I just ask, earlier in the day, you were kind of going in depth, kind of a science lecture on what exactly...
Nutrient cycling?
Yeah.
Okay.
So what we're trying to do here.
How do plants actually eat and consume their food?
Yeah, to explain that.
It's not simple. So plants have 3 methods. There's only 3 methods that a plant can use to absorb nutrients from the soil. Osmosis is one of them, but osmosis removes water from the plant, so they'll wilt over with osmosis. That's not their main mechanism. Active transport. So you have the cell wall, you have to have a channel that will recognize the nutrient the plant needs, and it takes ATP or energy to move that mineral from the outside of the cell to the inside of the cell. That's also not the main mechanism. It's used, but the main mechanism that plants use to eat or absorb nutrients from the environment is just called a diffusion gradient. So you have to have a large amount of nutrients in the local environment. And when the concentration is different between the inside and outside environment of the plant root, it's kind of like putting a spoonful of salt in just water. Over time, that salt will diffuse, and that it diffuses because it's a gradient. You got high concentration, low concentration. The same thing goes on inside plant root. Plants are looking for nitrogen, say, or phosphorus, or sulfur, or boron, or whatever the nutrient they might need. And they literally use everything from the periodic table. So if you have a high concentration of nutrient outside the cell, let's talk about nitrogen, and there's no nitrogen in the cell, that gradient will move the nitrogen from the outside through the cell walls to the inside. So it's going from a high concentration to a lower concentration in the cell. What the plant does inside the cell, inside that root hair, it attaches that nitrogen to a sugar. So now that makes it a protein, and then the plant will move that protein off to, do construction of the plant. So I don't know what do you call it. Tissues. It'll use that protein now to develop tissues that the plant itself needs. But,
cellulose?
Huh?
Cellulose?
Cellulose is one of them.
Okay.
But there's other, whatever a cell would need, it'll use those proteins to build its own body structure. But by moving that nitrogen now out of that local environment, it created another deficit of nitrogen. So now you have that gradient recreated. So got a new gradient, another nitrogen can move inside the plant through that diffusion gradient, and then that nitrogen will be attached to the sugar, and off it goes. So the plant takes sunlight, CO2 from the atmosphere, and water from the roots, and it creates sugars. That's all the plant does.
That's photosynthesis.
That's photosynthesis. That's all the plant does. Those sugars, about 80% of them it releases into the environment because it's farming. It's farming fungi and bacteria. Fungi and bacteria are the only two species on the planet that can degrade organic matter. A plant can't digest another plant. Plants don't produce digestive enzymes. They can't digest organic matter and use that as a food source. So why we put organic matter in the soil? It's because the fungi and the bacteria can break that down. Now when they break it down, fungi and bacteria also have the ability to go into this solid rock and extract nutrients through acids and enzymes. They're the only two species. No other plant or animal on the planet can do that. So by mining those minerals from organic matter or solid rock, the bacteria gets what they need. But the plant communicates with outside environment through these sugars that it creates. So the plant can create, I've heard up to 5 million different sugars.
Wow.
And depending upon what stress the plant is experiencing at any moment in time, through evolution or the DNA knows when I need this nutrient, I should produce this sugar and put that out into the environment. And what that does, it stimulates the growth of particular specific species of bacteria or fungi that has the innate ability to mine that nutrient that the plant needs from the environment. Now, the bacteria and fungi, well, let's say bacteria, don't give that nutrient to the plant. It keeps it for itself. So how does the plant get it? The plant made a sugar, it farmed and put out sugars that cause the bloom of specific bacteria that has the ability to get a nutrient from the environment. But how does the plant now get it? Well, the carbon to nitrogen ratio of bacteria is 5:1. But the predator, the protozoa, which can't mine minerals from the environment, it only eats bacteria, it has a body structure with a carbon to nitrogen ratio of 30:1. So that protozoa, the flagellate or the amoeba, it needs to eat 6 bacteria to get 30 carbons. 5, 10, 15, 20, 25, 30. But it only needs 1 nitrogen. So it got 5 excess nitrogens when it ate those 6 bacteria. So it poops out those excess nutrients, those 5 nitrogens, back into the environment. Now, if that happens real close to the plant roots here, it creates that diffusion gradient, and now that's how the plant can now reabsorb that nutrient that it needs. So it's like a 2 or 3 step process. The plant feels stressed, it puts out a sugar, it creates a bloom of specific bacteria or biology that can mine that material that the plant needs from the environment. The predators are amoebas, flagellates, and nematodes. They all eat bacteria, that's all they do. There is a root-feeding nematode, but they're rare in the environment. So anyway, the predators eat the bacteria. There's a difference in body structure between the 2 species, and the excess nutrients that the third trophic lever, the nematodes, the bacteria, and the nematodes, the flagellates, and the amoeba, they poop the excess nutrients back out into the environment, and then it becomes plant available at that point, and the plant comes over. So that's nutrient cycling, and that's what we're doing here. We're recreating nutrient cycling in the backyard. So
as part of the poop loop, right?
That's part of the poop loop, and the whole world runs on poop. Everybody's got a poop problem. Either got too much, not enough, can't get it, or can't get rid of it, you know?
Okay.
So, that's the poop problem.
So that's an awful lot about what looked like water being poured on mud.
It's like green water.
Right.
So the plants that you see in there is a food source for bacteria, and it's also a source of biology. So there's all kinds of species growing on all kinds of plants. So I went around the yard and I just picked up, little handfuls here and there and added to the bucket. And you want local biology because they can survive in your environment. If I took biology from, say, Florida up here to where it's really cold in winter, it may not survive. So you'll be working against yourself. So
one quick question. So we tested this soil, didn't have much life in it. We've applied this. Any estimate for
usually 1 growing season is all it takes. So we're putting this out in the spring. The response should be pretty good. Ideally, we would have put it out in November, let it get established. But really need to do this about a minimum of 3 times. And then the story is told about the Mayflower. The first group that came to the new land, they didn't make it, but they built some structures. And that's what I'm doing here. These are pioneers we just put out. A lot of them will die, but a lot of them will survive. But they're recreating and reestablishing a foothold.
And when you say a lot, how many did you count in it?
We put out billions and billions.
Billions and billions.
Yeah, we put out billions and billions, probably trillions actually. But each milliliter, 20 drops in a milliliter, had 1,560,000 flagellates in it. I mean, when I looked at the microscope, it's just teeming with them, with flagellates. And I made a video of it. I'll post that on the website, on the YouTube channel, so you can see what was in there. So we'll keep an eye on it and see how it worked out.
That's cool.
And we're not going to give up. We'll just keep at it till we're successful. So that's
all
how it
right.
works.
Thanks.